Okay, well, welcome everyone to Chapter 2, Part 2 of Chapter 2, where we'll be talking about the chemical levels of organization. Last lecture, we talked a little bit about how atoms are involved in the process of forming bonds, and how those bonds can be used to create macromolecules. And today we're going to be specifically talking about the macromolecules that are important for the function of the human body. So if you haven't watched the part 1 of this lecture for chapter 2, you might want to go back and watch it. And if you have, then you are ready to go here. So just a quick reminder in terms of what the expectations are for this week. So make sure you have your study guide for questions for chapter 2 prepared for Thursday. This is kind of the second section again for those particular questions. And then also make sure you've completed quiz 2 on chapter 2 by Friday at midnight. The adaptive practice is going to be due next Monday. So quiz 2 and homework 3 will both be found on Wiley Plus. So make sure you're going to Wiley Plus to go ahead and take these both quizzes and homeworks. So again, quiz 2, chapter 2 is due by Friday at midnight. Homework 3, adaptive practice 3 is due by Monday. Okay, so again, the purpose of this particular chapter is to introduce some of the fundamentals of chemistry and how chemistry pertains to the human body, talk about how matter is organized, the difference between weight and mass is something that we talked about last time. We talked about how chemical bonds were formed, and then today we're going to compare and contrast some organic and inorganic structures and talk about some macromolecules. So the study guide questions. So you'll notice the study guide questions for today's chapter 2 are different than the ones you completed for Tuesday's class. Here we'll be talking about things like pH and then all of the different types of organic molecules that make up cells. We'll go through each one of those different macromolecules, talk about some of their properties, what they're utilized in cells, and then we'll finish by talking about the molecule adenosine triphosphate, which I kind of alluded to last class, and it's important for cellular function. So last time we left off talking about different types of mixtures. Again, I just wanted to kind of overlap a little bit with where we finished with the last lecture. So a mixture, again, is a combination of elements or compounds that are physically blended together but not bonded together. Again, a solution is a type of mixture in which the compound has completely dissolved into solution. It's no longer visible. And then in a colloid solution, you're going to have some solution parts of the solution that are dissolved and some that have not. So you kind of have like a semi-visible cloudy mixture. The example given in the textbook and the example we talked about last time was milk. And lastly, a suspension is basically a liquid or slightly viscous solution in which you can see with your naked eye kind of visible chunks. I always think of if you've ever seen Jell-O and a Jell-O mold and maybe pieces of fruit within that Jell-O mold, that's a kind of a nice example of a suspension because you can kind of see the large macromolecule fruit pieces within that suspension. I think last time we also talked about percentage and molarity and how if you're working in a lab, these are very important concepts. Again, percentage is mass per volume, and if you were going to make up a percentage type of solution, let's say if you're going to make 10% sodium chloride, you take 10 grams of sodium chloride and put it into 100 mils of solution. If you were going to make a 10% solution of glucose, you would do the same thing. You would take 10 grams of glucose and put it into 100 mils of solution. So you're taking a mass, in this case 10 grams, and putting into a volume, in this case 100 mils. And so usually the 100 mils, 10 grams is the easiest way to do that, especially if you're making up, or if not, when you're making up a 10%. So if you had, let's say, a 5% sodium chloride solution, you would be taking 5 grams of sodium chloride and putting into 100 mils of solution. But I'm not going to be having you guys do solution making in this class, but if you are in a lab class, this might be valuable information. Again, moles or molarity is moles per liter. This is going to be based off of the atomic mass, and so you're going to need to know the atomic mass in order to do this calculation. So for sodium chloride, that is 58.44, and that goes into 1 liter of solution. And so, again, it just kind of depends on the solution that you're making with the atomic masses of that compound that you're dissolving into that solution and what the molarity that you're trying to achieve is. But again, not the specific focus of this course. So on a quiz or an exam, I probably wouldn't focus too much on these types of concepts. In terms of acid, bases, and salts, these are important for the human body, just because we do try to stay as neutral as possible, but there are areas of our body in which acids and basic environments exist. But so it's important to recognize exactly at the chemical level exactly what is an acid and a base. So an acid essentially is where you have a compound that's going to easily give up hydronium ions, or the H+, and that H+ is going to make that solution very acidic. Versus a basic solution where you have a compound that's easily giving up the OH, or the hydroxide molecule. Again, it's the presence of that OH, or the hydroxide molecule, that's going to make that a basic solution. So if you have a high concentration of H+, that's acidic. If you have a high concentration of OH-, that's basic. And then in the case of a salt, the example that you're seeing here is potassium chloride. You can see that you have the two ions that are formed, the potassium ion and the chloride ion, that are dissolving into that particular solution. Again, this is not a chemistry course. I'm not going to be asking you about the deeper chemical properties of this, but it is important that you recognize is that a acid is an H+ kind of heavy solution, basic is an OH- heavy solution, and that a salt has those two ions that are dissolved. And so in the case of, let's say, sodium chloride, let's see, in the case of, let's say, sodium chloride and NaCl, if that was to be dissolved into a salt solution, you would have Na+ going in there into it, and then that Cl-. So those are kind of, again, another example of a salt. I think when we think of salt, often the salt that we think of is a sodium chloride. And so potassium chloride is also a salt, though, because it is an ionic substance that's being dissolved into a solution. Okay. So again, in terms of pH and kind of recognizing what pH is and how it matters, again, you can see here that the concentration of OH- is much, if you have a very acidic, you have a very low amount of OH-, and then if you have a lot of H+, in this case 10 to the 0, you have a very low pH. So in this case would be the pH of 0. And so as the amount of moles per liter essentially increase, you get it closer to that 10 to the 0 power, you have a far more basic environment. So lots of H+ means that you're acidic, lots of OH- that you can see like right here with the 10 to the 0, very, very, very basic. If this nomenclature, this scientific annotation isn't, let's say, very familiar to you, the 10 to the -14th basically means that there are literally 14 zeros, I guess 13 zeros, that are given this concentration. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13. So that would be the concentration of OH- here. And then if we're looking at the very concentrated, there would be, in the case of 1 moles per liter of OH- here, versus here where you're having this number moles per liter is the again, is how we're annotating this. So you have got far fewer OH- at this end of the spectrum, and here you have far more, and so that's going to lead to the very basic. So if you ever have any questions about kind of the difference between acidic and basic, I think this is a really good slide to come back to, because it kind of just shows you the shifts that occur. What's interesting to kind of note on this particular slide is that where you see right here, with your neutral substance at pH 7, you've got equal amounts of both hydroxide and hydronium ions, and so that's what makes it neutral. Those two kind of essentially balance each other out, and so you have a neutral environment. And we talk about physiological substances. We most often operate at physiological pHs, which is right around that pH 7. There are a couple different exceptions to that rule where you don't see that pH 7 followed. And again, like I mentioned before, there are a couple different environments within your body that you don't want to have a neutral environment. You need acids or you need bases in order for that area to take its function. For example, I think we'll talk about this here in a second. A great example of that would be the stomach, right? Your stomach, it needs to break down the food that you consume, and so that's a very highly acidic environment, meaning that there are a lot of hydronium ions there helping assist the breakdown of those proteins and carbohydrates and different things that you're consuming. So this is, again, is a great example of some of the different areas that we see in of different pHs within the body. So you have things like gastric juice, again, like I mentioned before, it's a very acidic environment. So a pH of 1.2 to 3, that means we've got a lot of H plus there. Lemon juice, a vinegar, carbonated soft drink, orange juice. Shockingly, vaginal fluid is also a very acidic environment. So again, these all kind of fall under the acidic category because they've got a low pH. So low pH means that something is acidic. So coffee and urine, again, all falling very much under that acidic nomenclature. If we look at some of these other kind of physiological substances that we're using here, something like saliva, no, it's actually well within that kind of neutral pH-ish area, still kind of low on the lower end of being acidic. And then here you can see some of the more basic solutions as we raise in our pH. So these would all be considered a little bit more basic, meaning that they have a high pH and they're going to have lots of OH minus available, and that OH minus available, and that's what's going to essentially create the acidic environment. And things like blood is just a little bit in that direction. Semen, cerebrospinal fluid, pancreatic bile, milk of magnesia, lye. But I mean, with that being said, kind of you'll also notice that I'm saying these things are basic, but they're very, very subtly basic. They also kind of could be characterized as maybe neutral because they are very close into that 7 pH range. All of these are kind of, you know, in that 7-ish range, very kind of neutral area. And pH is important because it aids in protein stability. We'll talk about proteins a little bit more in this lecture. Proteins are kind of the building blocks for most cells and assist the other macromolecules in functioning. And so if you have, let's say, a protein in a highly acidic or a highly basic environment, it's going to affect the way that protein functions because of the way it folds. But we're not going to get down to that level of understanding here. But yes, basically, pH is very important for protein function, for proteins, but other macromolecules as well. So this animation here is showing the acids and bases, pH and buffers animation. Feel free to click on this and it should shoot you towards the Wiley Plus. I found out last lecture that if I click on this and it sends me to another website, you're not able to hear it. So I'm not going to do that for this lecture. So please make sure that you're taking the time to open up this lecture in PowerPoint on your computer and then click on those links. So buffer systems, again, because we want things to stay neutral. Maintaining homeostasis is so important for the body that it doesn't want to deviate in great degrees from that neutral pH of 7 in most places, not all places, like I said. So there are buffer systems that are set up in order to maintain that neutral pH. So this converts strong acids or bases into weak acids or bases, meaning it makes them far less likely to either donate a hydronium ion or donate a hydroxide ion. It makes them kind of hold on to them a little bit more tightly. It's probably the easiest way to say it chemistry-wise. They just kind of hold on to them a little bit more tightly and aren't as susceptible as giving them up to create kind of those random H pluses and random OHs kind of floating around in the environment that would make it acidic or basic. Okay, so that kind of wraps up the real kind of individual atoms portion of this lecture. Now we're going to talk about an overview of organic compounds. Now I was looking over this lecture and recognized that they don't really divide it up into the way that I'm about to tell you now, but it does, I think, kind of make it a little bit easier to think of the fact that there are 4 major macromolecules that make up cells. And so those 4 macromolecules you'll start with, we'll just start with sugars. And sugars are sometimes also called carbs or carbohydrates. And we'll talk about the structure of those. Often sugars or carbs or carbohydrates have kind of a circular cyclical nature to the chemistry behind them, and we'll see those. They're ring structures. Next you have lipids. Lipids can also be called fats. We'll probably interchangeably call them lipids and fats. Lipids are different in chemical structure. They usually have kind of a single carbon backbone and then these long kind of legs extending off of them, and then we'll talk about that structure a little bit more. So we have sugars, again, that are kind of cyclical in nature in terms of their chemistry, and that's probably the worst sugar cyclical drawing that you've ever seen. So you've got sugar, lipids. We'll talk about proteins. I keep kind of alluding to proteins. Gosh, this handwriting is terrible. Proteins are kind of the building blocks of cells. They do a lot of the functions. We'll talk about enzymes, which are a type of protein. So proteins, that's a B. And then lastly, we'll mention kind of DNA and maybe mention a quick bit about RNA, deoxyribo, and ribonucleic acids, which are going to be the genetic material for cells. So kind of the overview of organic compounds of 4 macromolecules we'll talk about today are sugars, or also called carbohydrates, lipids, which are also called fats, proteins, and then DNA and RNA are the nucleic acids. And each one of these has a very specific function in your cells. But if you break your cells down into like the major macromolecules that help assisted in carrying out its function or compromise or make up your cells, these 4 macromolecules are basically it. So there's are very important molecules that we're going to just talk about today, and it kind of helps us make that transition from talking about individual atoms and molecules into kind of the larger structures that comprise cells. Sorry, there's a little bit of delay here. So again, we talked a little bit about the difference between organic and inorganic compounds. These being organic compounds, they're all going to be comprised of carbon. Carbon is one of the most important elements in cellular function in creating these macromolecules. And then last time we talked a little bit about why that is, because carbon does have those 4 electrons hanging out in the outermost shell of its or orbital of its electron shell. They can always form those 4 bonds with other types of atoms. Those other atoms that we most often see them bonding with would be hydrogen or oxygen or nitrogen. But carbon can combine into a variety of shapes. So it's kind of like when you have let's say a set of components that you're building, this would kind of be that backbone component that you can decorate with a lot of different components and make it a really interesting in different ways. Carbon compounds do not dissolve easily in water, which would make them polar. And so when we talk about things like lipids that have a ton of carbon in them, that's why lipids don't dissolve easily in water. But carbon compounds are also a good source of energy. So when we talk about things like lipids and fats or carbs or sugar, breaking down those bonds can make them a great source of energy, and that's why we consume those things on a regular basis. So when we talk about these carbon molecules, like I said, you've got that standard kind of carbon backbone, but you can kind of decorate that carbon backbone with a variety of other structures. And so these next few following slides kind of show you some different structures in which you can decorate that carbon backbone. So you can decorate it with a hydroxyl group, which is like an OH group. We talked about that just a second ago when we were talking about basic solutions. You can decorate it with a sulfohydryl group. or this has a sulfur within it, a carbonyl group, and ketones contain a carbonyl group within the carbon skeleton. And again, it's the kind of chemical properties of each one of these that can add to the properties of that larger carbon structure. You can also have aldehydes, carboxyl group, and ester group. The carboxyl group is important when we get to talk about proteins. It's part of the basic protein structure. And ester groups. So these are just basic chemical groups. Again, since it's not a chemistry course, I'm not going to show you these particular side chains and ask you to identify them. This is just kind of more of an FYI. These are some of the ways in which the carbon backbone can be decorated. You can also decorate it with a phosphate group. We see phosphate groups, particularly when we're talking about the nucleic acids. Both DNA and RNA have phosphate groups as part of their backbone. An amino group, just like we talked about the carboxyl group a second ago being important for proteins, the amino group is also important in protein structure as well. And it helps helps the bonds form between additional amino acids as they're creating a growing protein structure. Okay, so the 4 macromolecules we talked about here are sugars, fats, proteins, and nucleic acids. So the first one we'll talk about more is sugars, or carbohydrates. So like I said in the introduction, these sugars are ring structures. So glucose is an example of a ring structure. Again, if you're not a huge chemistry buff, you can see that it's a ring because it's circular, right? You've got a circle of some sort kind of drawn here. And you can see that each one of the kind of edges of this circle are carbons, with the exception of this oxygen that you see here. And some of those carbons have larger structures kind of hanging off of them, which kind of gives you the identity of glucose versus some of the other sugar structures that we'll be looking at. You can see the standard shorthand given over here. That's maybe a little bit more simplified. Instead of drawing out that entire structure that you see up here, it's given in more of a shorthand version here, CH2OH, because there's 2 H's here and an OH here. So the shorthand version of it is like this. But again, this is not an organic chemistry class. I'm not going to ask you to recreate any kind of structure, because I really don't think that that should be your focus of an anatomy and physiology class. But you can see that there is a ring structure for this particular sugar. When we talk about different types of sugars, like I said, that's the basic sugar ring structure they saw for glucose. The reason that we probably gave you that as an example is because it's the main blood sugar. Fructose, galactose is in milk sugar, deoxyribose is found in DNA, ribose is found in RNA. And the fact that they're monosaccharides mean that they're comprised of just that one single ring structure. Disaccharides, things like sucrose, lactose, and maltose, means that they're essentially, instead of just that single ring structure that comprises them, you're going to have 2 ring structures. So mono meaning 1, di meaning 2, and so in the case of the disaccharides like sucrose, lactose, and maltose, those are all considered disaccharides. They've got 2 ring structures that comprises them. Again, 1 ring structure mono, 2 ring structure di, and we can see examples of those different types of carbohydrates. Polysaccharides, meaning maybe not just a single or a double ring structure, but these are huge chains of ring structures. Poly meaning just multiples. Glycogen, which is formed in the carbohydrates of animals, starches, and cellulose. Starches we eat and consume, again, something that we consume, and this is a high energy source. Of those bonds that the carbon structures have are going to be a source of energy for us. And then cellulose is in plants and cannot be digested in humans, just because the bonds that are created between those ring structures, our body doesn't make enzymes to break those bonds down, and so because of it, we're unable to digest those. So it's interesting to think that, kind of evolutionarily wise, we have to have enzymes to make or break these bonds. So if there's a chemical or a structure that doesn't have the right kind of orientation for us to have an enzyme to break it down, we're unable to digest it, as in the case of cellulose. So here's the actual chemical structures of each one of these. You can see ribose and deoxyribose. These are all monosaccharides, just a single ring structure. Deoxyribose and ribose are both important for the nucleotides. So ribose would be in the basic composition of RNA, and deoxyribose would be in the basic composition of DNA. And when we get to talk about these structures, you'll see exactly where. And these are all considered pentoses and hexoses. Hexoses would be glucose, fructose, and galactose, and those are sugars that are most often consumed for the breakdown of energy. When we talk about metabolism, we'll go back and we'll see glucose many different times, because we'll see how glucose is actually metabolized by cells and how this ring structure slowly but surely loses carbons, and each one of those carbons is going to give off energy for the cell. So again, glucose is going to be one of the largest sources of energy for cells. We'll talk about how that carbon, once that ring structure gets broken down, each one of those carbon bonds gets broken, energy is released for the cell. And that's probably one of the most basic ways you can describe cellular metabolism. Okay, so like I said, those are monosaccharides. These are going to be the disaccharides that have the two ring structures that are joined together with this particular bond, this what they call a glycosidic bond. This bond here, we're seeing can combine galactose and glucose, or glucose and glucose. Now, when these ring structures are combined like such, this becomes nomenclature-wise maltose, and a galactose and glucose combined becomes lactose. So it's just, again, an organizational nomenclature where you've got multiple compounds that are combining to become these more complicated structures. Okay, polysaccharides, long chains. Here you can see an example of a glucose monomer within that large, just extended chain of polysaccharides. And there are many, many glycosidic bonds. Glycosidic bonds are the bonds that form between these sugars to create these polysaccharides. Okay, so we started by talking about sugars or carbohydrates. Next, we're going to start talking about lipids, or sometimes more commonly called fats. So lipids in the body. So like I said, kind of the basic structure of a lipid is to have a carbon backbone and then have these long tails kind of extending off of it. And so we'll talk about each one of these and kind of what the structure of them looks like. So fatty acids is probably one of the more commonly seen type of lipid in the body. They use triglycerides and phospholipids to generate adenosine triphosphate. So again, if you remember anything from basic biology, ATP is energy for the cell. It's generated in the mitochondria, which is the powerhouse of the cell. And we'll talk about that process a little bit when we talk about metabolism. And then triglycerides in the body are used for protection, isolation, and energy storage. And then phospholipids are the major lipid component of cell membrane. Phospholipids are really interesting because they have the differences in charge, which are considered amphipathic, amphi meaning both. So if you think of amphibians, amphibians can be in both water and on land. An amphipathic molecule can be both water-loving and water-hating. And so phospholipids can actually be both of those things, because they have to basically interact with both water aqueous environments like outside of the cell and inside the cell, which might be a little bit more well, I guess because phospholipids branch in together, they have the water-loving part on the outside and the water-hating part kind of on the inside. And I think we're going to be talking more about that. Steroids are also an example of lipids within the body. Things like cholesterol, bile salts, and vitamin D, all steroids that are important for cell signaling, but also sometimes cofactors for biological processes as well. So just like sugars are important for the function of the body, lipids are also important. Last one down here, that vitamin D helps regulate calcium levels in the body, needed for bone growth and repair. And there are also other hormones that we require, adrenocortical, sex hormones. Again, these hormones are signaling over very long distances within the body. And other lipids, things like keratin and vitamin E, are important for other bodily functions. Things like carotenes are needed for the synthesis of vitamin A, also function as antioxidants. We'll talk about an example of where you can actually get vitamin A and then also vitamin E, which prevents wound healing, prevents scar tissue, and so on and so forth. So these are all examples of lipids that have a very specific function within the body. Very last slide here for types of lipids, vitamin K and lipoproteins. And the vitamin K is going to be required for blood clotting proteins, and lipoproteins transport lipids in the blood. Okay, so the structure that I kind of keep coming back to for fatty acids are that backbone with those long legs hanging off of it, and that's what the kind of way I keep drawing it. And so here you can see an example of that. So fatty acids can be saturated or unsaturated. So when we're talking about saturation versus unsaturation, so think of a saturated fat being like this that you can see here. The glycerol fatty acid creates this palmitic acid. You've got this saturated fat, and the reason it's saturated is because it's essentially being saturated by hydrogens. So every carbon that you see here has a hydrogen that's been bonded with it, and so it creates a very straight leg of that fat. If you see here the example of this palmitic acid and this oleic acid is monosaturated. This is a unsaturated fat because it's got this double bond here. So this double bond actually causes a kink in this leg of this lipid, and that kink causes it to kind of kick out to that side. So you can see it's kind of not completely straight like this palmitic acid here. So it's completely saturated with hydrogens, and this one is missing a hydrogen kind of here and here, and because of that we have a double bond, and the double bond causes a kink. So this is unsaturated. So this is unsaturated with a kink, and saturated with hydrogens means that it's straight. So when we talk about lipids and fats, if you probably often hear the terms saturated and unsaturated fat kind of used to describe foods. The more solid a fat is, so if you think about a stick of butter, butter is composed of saturated fats because it's got these very straight bonds that are composing it, so they can pack very tightly together, and that makes it solid. Versus unsaturated fats, where you've got these kinks that are occurring, right? And because of these kinks, there are much greater spaces between those legs, and because of those much greater spaces, they're actually liquid in nature. So if you think about oils, oils are types of unsaturated fats versus butters, where that is a saturated fat. So it's a good example of the difference between saturated and unsaturated fats. The second panel here is just showing a dehydration synthesis involved in making a fatty acid. So remember last time we talked about different types of bonds. The dehydration synthesis that occurs here is you have this OH group that is lost with this H, and that gives off water, so you have dehydration that's occurring, and this fatty acid molecule gets kind of attached to this backbone. So this glycerol molecule that you see here, and this fatty acid that you see here, essentially create a new molecule. Okay, so saturated and unsaturated fats, and then the dehydration synthesis to create a lipid. Okay, so hopefully, if you do have any questions, feel free to post them onto CoreSite or to email me if they're arising as you're going over this lecture. Okay, so these are fatty acids. Triglycerides have tri, meaning 3. They provide protection, insulation, and energy. Triglycerides have those 3 legs hanging off of them, and you can see here with the addition of each one of those legs, dehydration synthesis has occurred, so you're gaining, or I guess all water is being lost through that, through the addition of that leg. So you can see there's palmitic acid, stearic acid, oleic acid being joined to that glycerol backbone through an ester linkage, and this is monosaturated unsaturated. This is saturated. This is saturated. You can tell that they're saturated because they're saturated with hydrogens, and so there's no double bonds, no kinks in those legs. Same thing here with stearic acid, completely saturated with hydrogens, no kinks in those legs. And here you can see with oleic acid, oh, there's a double bond. That double bond, because you are essentially kind of missing two hydrogens here, causes that kink in that leg, and that's going to lead to that unsaturated fat. But that's the composition of a triglyceride. It's got those three different legs hanging off of it. Each leg is attached via a dehydration synthesis, and those legs that are attached can essentially be comprised of saturated or unsaturated fats. Okay, I alluded to the fact of phospholipids, how they're amphipathic. And so when I said amphipathic, remember amphipathic. Again, you think about an amphibian. Amphibian loves water, loves H2O, and or can love water or be a water hater. So amphibians can be in water and not be in water. Now, if we look at this phospholipid here, we have what's called a polar head group. If you remember last time, polar molecules actually love water. And then if we look at this fatty non-polar tail here that's comprised of both the saturated and unsaturated fat, this actually hates water. So the way phospholipids work, again, if you have a water-loving and a water-hating area, what it's going to do is it's actually just going to create that phospholipid, what it's called a bilayer, meaning two. It's got two phospholipids that compose it on each side, or one on each side. So the water-loving heads are sticking out, hanging out here with all the water. They love being outside. And then this is also facing inside of the cell, which is also comprised of water. And then it's got this nice fat layer. So you can think of each one of your cells actually has this nice fat layer around it. And the reason it does is because this fat layer is hard for other polar molecules to come through. So it helps create a nice fatty selective layer for the cell. And so not everything is able to enter. It's a little layer of fat protection. So other polar molecules can't just enter the cell freely. So that is the phospholipid bilayer. Steroids, as we introduced them as well, are signaling molecules that can travel long distances within the body. Here you can see steroid cholesterol, estradiol. Cholesterol actually acts as a signaling molecule, but also acts as a molecule that comprises our phospholipid bilayer, or our layer around the cell. Estradiol is estrogen, a signaling molecule in females and somewhat in males. Testosterone, also found in both sexes and is a signaling molecule in both. And then cortisol, stress hormone that is released. And we'll talk more about these hormones when we talk about their function. That might be a topic that's covered in semester 2. Okay, so we have talked about lipids. We've talked about, sugars and carbohydrates. We've talked about lipids / fats, and now let's talk about proteins. So proteins, I would say, are kind of like the building blocks of the cell, kind of the major backbones and structures of the cell, carry out a lot of the legwork of the cell. So fats and sugars both provide energy. Proteins are kind of like what makes the house of the cell outside of the phospholipid bilayer, and then we'll talk about DNA being the blueprint for the cell. So proteins give structure, regulate processes, provide protection, assist in muscle contraction. We'll talk about that a lot. Transport substances and serve as enzymes, and we'll also talk about enzyme function within this lecture. Okay, So in terms of this function of proteins, structural. So again, they provide structure within the cell, but also within different tissues within the body. So if we talk about collagen and bone and other connective tissues, keratin and skin, hair and fingernails, those are proteins that give both our body and our individual cells the structure and kind of holds everything in place in order for those cellular functions to take place. Regulatory, so proteins can actually serve as receptors, control growth and development. You also have neurotransmitters that are composed as proteins. So the hormone insulin, the neurotransmitter known as substance P, also are going to be considered regulatory proteins. Contractile, so when we talk about the shortening or lengthening of muscle cells, myosin and actin are important proteins that kind of regulate that process of muscle contraction and allow the fibers to kind of contract and to, once they release, are going to relax. Immunological, things like antibodies and interleukins. So when your immune response gets activated, you have proteins that get released that allow you to identify non-self substances, and these are called antibodies. And antibodies are part of the way our immune, again, recognizes self from non-self. Transport. You can think of proteins in aiding this movement of substances from one place to another. So if you have any molecule that needs to move into or out of a cell, into or out of a tissue, and it has to happen selectively, there needs to be some type of protein kind of bridge between those two areas that allows for that, because the protein, what it's going to do is hopefully regulate the passage of that substance into and out of the body. It can also help regulate movement across long distances. So the example that's given here is hemoglobin, which is going to be a protein that transports oxygen to all of the tissues within your body. And lastly, what's mentioned here is the catalytic function of proteins, and proteins act as enzymes, and enzymes have an important role in lowering that activation energy that allows reactions to take place. So if we go back to the first lecture of chapter 2, remember when we talked about activation energy? Well, enzymes lower that activation energy, so it requires less energy and optimizes the environment for a reaction to take place. Okay, so what are proteins composed of? So proteins are made up of amino acids. So there are 20 amino acids. So all of the proteins in your body are comprised of the same 20 amino acids. And those amino acids are kind of joined together through a series of peptide bonds. And so here you can see an example of an amino acid. So let's just start here where you've got a carbon here, and that carbon is going to be decorated with what's called a side chain. That side chain, like I said, there are 20 possible side chains here. So there are 20 possible R groups that could possibly be attached here. But all this other component of the protein looks exactly the same for all of the amino acids. So you've got a central carbon, you've got a carboxyl group on one side, you have an amino group on the other, and it's these carboxyl and amino groups that are going to essentially form a kind of chain link to the next amino acid. So it's like if you've ever had a thing, adapter or connector that has kind of like a male and female side, like a knob and a tube, and you connect it, and then a knob and a tube, and you connect it, then you can connect them all together. Proteins are kind of the same way. And here you've got an amino group in your carboxyl group, and you can actually form a bond between this carboxyl and this amino group, and that's going to form your peptide bond. So like I said, you've got 20 different possible side chains here. So the example that is given here is you've got glycine. Glycine is the simplest amino acid. Its side chain is just a hydrogen. Here it is an example of a different side chain. Again, these are both the R groups. This one is cysteine, so you can see that the side chain is a little bit more complicated. And then here's another, side chain, tyrosine, or another side chain, lysine. So each one of these side chains decorates the side of the amino acid, but you can see that this backbone here remains the same between all of them, right? You've got an amino group, you've got a carbon, and you've got a carboxyl. You've got an amino group, you've got a carbon, and you've got a carboxyl. And so this kind of repeated pattern here allows you to link these things together again and again in a very repetitive pattern. It's worth also noting that these side groups or these side chains have different properties. Some of them, are more polar or non-polar, and those properties are what allow them to carry out their functions or create different types of structures. So again, this isn't a cell biology class, so if you have questions about this, I'm super happy to talk more to you about it, but I don't want to get too much in the weeds here, which I'm sure you're already like, oof, chemistry, this is not fun. Okay, so drawing the peptide bond. So like I said before, you've got your amino group, you have your carbon, you've got some whatever side chain you'd like here. In this case, it's an H, so this is glycine. You've got your carboxyl here, and here's your amino group, your carbon, your side chain, in this case for alanine, and then you've got your carboxyl group. And you'll notice that the carboxyl and the amino groups join here, and you're losing water. You're losing H2O. So we know from lecture 2 that this is a dehydration synthesis when you lose water, right? And then the loss of water allows you to create what's called a peptide bond between those two amino acids. So amino group, carbon, C. So this double bonded O kind of pops up there. You see it right here. And then there's a bond that gets formed between this carbon and then this nitrogen. You still have this H hanging out. But again, you've lost that OH from that carboxyl group and that hydrogen from that amino group. Again, that's given off as water. And then you have the peptide bond formation. Still have the amino group and carboxyl group intact on each side. So this is peptide bond formation and how you can just link together hundreds upon hundreds of amino acids. And each one of those amino acids, again, you just link them together. You continue to grow that chain. That chain gets longer and longer and can eventually kind of wrap upon itself and create a three-dimensional structure that has a very specific function. So here's kind of what that looks like. You can see you've got your very, very simple list of like, here's an amino acid. Oh, here's amino acid 1, here's amino acid 2, here's amino acid 3, kind of that list of amino acids you can see here. That's the primary sequence when you just kind of list out the amino acids. And then once it folds in upon itself, in this case you can see it's formed an alpha helix. An alpha helix looks very much like a spiral staircase. If you've ever seen a spiral staircase, that's what the alpha helical structure looks very much like. In protein structure, you can either have that spiral staircase alpha helix, or you can have what's called a beta sheet, which is much more flat in terms of its structure. And then that's the secondary structure. Now that secondary structure, let's say you have like that twist, and then that twist and twist again. That twist and twisting again, like if it folds over on itself, that's called tertiary structure that you can see right here. So there's an alpha helix, there's an alpha helix, and this is considered the tertiary structure. You can also write it as kind of 3, this is 2, this is 1. So this is primary, this is secondary, this is tertiary. And then if you take these tertiary structures and kind of combine them, you can see that there can be quaternary structures. So it's like each kind of separate unit or subunit of protein comes together for a specific function, and that is called quaternary structure. Probably the greatest example for quaternary structure is we talk about hemoglobin. Hemoglobin is a protein, but it has 4 different kind of subunits that makes up that hemoglobin protein. And so those 4 different units coming together to actually kind of carry out the function of transporting oxygen, that's the quaternary structure of hemoglobin. Okay, so we talked about kind of the structure of proteins. Some of the functions, for example, would be enzymes or enzymatic function of proteins. So proteins and enzymes within the cell have very specific functions. Each enzyme must interact with a specific molecule. They're very efficient. What they're going to do is kind of change the conformation of that other molecule, and enzymes can be subject to cellular controls because enzymes are kind of carrying out these reactions. Some reactions you want turned up, some reactions you want turned down, some of you want those reactions you want to happen under very specific circumstances, and so other cellular controls will help those enzymes. And if you continue to take biology classes and you take a class that talks about like let's say biochemistry and cellular pathways, you'll recognize that there are a ton of enzymes, and there are enzymes that regulate enzymes that regulate enzymes. There are a lot. So this is a great example or cartoon showing you exactly what an enzyme does. So an enzyme, here you can see, is its own little protein globular structure. So this enzyme has what's called an active site, or like if you have, let's say, kind of a pocket in which another molecule can kind of fit within. So that other molecule comes and fits in that pocket, and the enzyme kind of moves a little bit to kind of alter its shape, and then the reaction takes place. And then once the reaction takes place, the protein or the enzyme kind of opens up, and that molecule leaves, and it's open for business. Another set of molecules can come in, and reaction can take place, and the enzyme will open up, and that molecule will leave. And so that thing that comes in is called a substrate. The substrate comes in, the reaction takes place, and then it opens up and it leaves. Now you'll notice that as this opening and closing happens, that the enzyme itself, it moves, but it doesn't change in composition. It's just opening and closing, opening and closing. So its composition stays the same, but the substrate, the things that are coming in, they're chemically changing their structure, they're reacting. And so the enzyme causes a reaction and alteration of the structure of the substances, but the enzyme itself just kind of opens and closes, but doesn't necessarily change in its chemical structure. So the enzyme kind of just helps those substrates react and facilitates the reaction of them, and because of that, it can rapidly help the cell carry out different types of processes. So the last picture I showed you was kind of a really simplified cartoon, obviously. If we look at kind of what it more looks like in the biological sense and the atomic structure sense, here you can see the same enzyme that's represented here. So this enzyme is represented by this space filling model that you can see here. Looks like just kind of this U shape. Should choose a different color. This space filling model, it's a U shape that you can see here, and you have a substrate that comes in that's glucose, and the substrate binds to that pocket, but you can see how this large, you have this large gaping hole here, and here it's much smaller, and that's because once that substrate binds, it causes what's called a conformational shift, which is a very sciencey way of saying the enzyme moves. The enzyme moves, and the reaction takes place, and then once the reaction takes place, the enzyme moves back, and the substrate ends up getting released. So yeah, sometimes scientists have nomenclature to make things far more complicated. So throughout this process, if you ever kind of hear me going down the nomenclature rabbit hole and just saying words that you're just like, I have no idea what that means, please just pop me up a message and say, hey, you use this term in lecture, what exactly do you mean by it? Another opportunity to kind of look at enzyme functions and how enzymes are important for ATP production, or sometimes enzymes are actually require ATP for their function to take place. So it's definitely worth clicking on that and checking out the enzyme functions in ATP. Okay, talks about lipids and fats, talked about sugars or carbohydrates. We talked about proteins, and kind of the last macromolecule we'll talk about today are nucleic acids. Okay, so everything else kind of comprises the cell, makes up the structures of the cell, carries out the functions of the cell. Nucleic acids are basically the blueprints to create all of those other things. So you can think of nucleic acids as being like the writing that allows the cell to take, okay, I've got this writing blueprint, how do I make this protein? I've got this writing on the wall, how do I make, you know, this protein that's going to have this enzymatic function, or how do I take this writing and make a protein that's going to actually help this enzyme make this lipid or fat? So it's really kind of the like the head department in the cell. If you thought, sometimes I like to think of the cell as a little protein making factory, you know, because if we look at the organelles, which we'll talk about a little bit more in depth, kind of as a protein making factory, the nucleus is kind of the headquarters or the R&D, like, okay, what are we going to make? What is the development here that's going to have the blueprint? And the nucleus contains nucleic acids, and that tells you everything else that that cell needs to be made. Okay, so DNA is that basic, is just that, oh, I'm like, why is it red? It's blue. Is going to be the genetic code that regulates most of the cell's activities, and then RNA guides protein formation, and there are many different types of RNA that carry out cellular functions, and we'll talk about maybe some of those. Okay, so in terms of DNA, I feel like you guys are getting a crash course in a cell biology class right now. So again, if you'd like me to go over any of this more in depth, please let me know, or less in depth. So for a nucleotide, that is comprised of a pentose sugar. We talked about that when we were talking about sugars. That's a monosaccharide, right? Because it's a single ring, this pentose sugar. If it is DNA, it's deoxyribonucleic acid. If it is RNA, it is ribose, deoxyribose, ribose. And then part of that backbone is you have a phosphate group attached to it on one side, and on the other side you have what's called a nitrogenous base. And this nitrogenous base is going to change depending on the identity of that stretch of DNA. So you can have this, the cysteine, thymine, or uracil that get added as a nitrogenous base. We'll just call this kind of like X, and X can be either one of these things, or X can be adenine or guanine. So when you look at a stretch of DNA or the code of DNA, you'll see represented by these letters, A, T, C, or G, right? These letters each represent one of these nitrogenous bases. So it's adenine, thymine, cytosine, and guanine in DNA. So these are your DNA molecules. In the cases of RNA, you know something is a stretch of RNA if it contains uracil. Uracil is only found in RNA, and in DNA, you only find C, T, A, and G. And in DNA, you'll find thymine. In RNA, you'll find uracil. So when DNA and RNA interact, thymine gets substituted by uracil, essentially. Other kind of, this slide has so much information on it. So again, ATCG make up the nitrogenous bases, cytosine, thymine, uracil, guanine, and adenine. Other things to note is that if we're going to kind of cluster these things together in terms of how their ring structures, you'll see these single ring structures here of C, T, and U. These are pyrimidines. Adenine and guanine are purines. And in terms of just memorizing the pyrimidines versus the purines, I won't have you do that for this course again, because I think that that's kind of more outside of the scope, but in case you're taking a biology course and you need to memorize them. The way that I do is that a pyramid to me kind of looks like a diamond. We were going to do a diamond ring. Kind of looks like a diamond, I guess. And what do you do to a diamond? You cut it, C-U-T, so cytosine, uracil, and thymine. So you cut a diamond to me looks like a pyramid. Again, just kind of weird name associations, whatever works for you. And then if I ever need to figure out what the purines are, I just know it by process of elimination. Yeah, so we already talked a little bit about how DNA is deoxyribose and RNA is ribose. Okay, So I think we've covered, again, this slide has so much information on it, and I could probably talk about it all day, but I do not want to go into too much detail because, again, this is not a chemistry or a biology class. I just want to introduce the ideas that DNA is comprised of these nitrogenous bases. They bind, create bonds with these pentose sugar backbones, and then there's a phosphate group hanging off of it. Now, these, we'll see if the next slide goes into it, or else I don't want to get lost in the weeds. So those sugar phosphates create these repeated patterns of bonds. Again, you've got a phosphate, you've got a sugar, you've got a phosphate, you've got a sugar, and that's what creates this backbone here. It's a a sugar phosphate backbone, and you've got your nitrogenous bases here hanging out. In this case, you've got thymine and cytosine on one side, and then on the other side, you've got your sugar phosphate backbone, and you've got your other two nitrogenous bases, adenine and guanine, and they're creating kind of the other side of this. Now, the stretch of DNA is a alpha helix, much like we saw with protein, and it looks like a staircase, right? This looks very much like a spiral staircase, and you can see kind of the stairs of the staircase are these pairs of nitrogenous bases. So the nitrogenous bases, what you would say, base pair with one another or create bonds with one another. So you've got T that base pairs with A, and you've got C that base pairs with G, and that's always the case. A base pairs with T, C base pairs with G. That's always the way that they base pair up, or they bond together. And you'll notice that the difference between those bonds is you, in the A and the T, you've got 2 hydrogen bonds. In the cases of the C's and the G, you have 3. So 2 hydrogen bonds versus 3. So again, this slide has a lot of information on it. You can see kind of the double helical structure that you probably have seen many times when you hear people talk about the structure of DNA. And so kind of the stairs of that staircase are those base pairs forming hydrogen bonds with one another. Okay, this is a lot of information. So again, if you have questions, reach out, post questions. I'm more than happy to have longer discussions or clarify this, just because I recognize this is a lot of information very quickly. Okay, so DNA base versus RNA. So DNA has a nitrogenous basis A, T, C, G. And for RNA, instead of T, you have U. So if I showed you like a stretch of, I won't do this, if I showed you kind of like a whole stretch of DNA or RNA, and there's a U present, and I said, is this DNA or RNA? You'd say, this is RNA because there is a U. If I showed you a whole stretch of like letters and there was a T present and no U's, you would say that's DNA because there's a T present, there's thymine present. The sugar in the nucleotide, again, those pentose sugar backbones that are monosaccharides. DNA is made up of deoxyribose, RNA is made up of ribose. In terms of the numbers of strands of DNA and RNA, this is a little simplified here, but DNA generally is a double-stranded structure forming that spiral staircase. RNA can be a single, and there are some cases where it kind of doubles up on itself, but we'll just say a single strand structure for right now. Here you can see the nitrogenous base, the number of bonds, the hydrogen bonds that form between them. So in A and T, you have 2 hydrogen bonds. With G and C, you've got 3. With A and U, you've got 2. With G and C, you have 3. And then how is it copied? Well, DNA is self-replicating, and for RNA to replicate or make more copies of it, it needs to use DNA as a blueprint. But again, this is not a cell biology class. I'm not going to have you memorize things to that level for this particular material. You're like, when do we learn about muscles? When do I have to memorize bones? Why am I learning about DNA and RNA? And it is important. It's just kind of the foundation of the rest of the steps of this course. Okay, so DNA versus RNA continued in terms of its functions. DNA's basic function is to just carry the information for making proteins. And it can be found in the nucleus, but it also is contained within the mitochondria. And if anybody wants to ask me why there's DNA in the mitochondria, that's also another really fun conversation kind of about evolution. And then RNA is crazy multifunctional just because it's got, a single strand of it, so it can actually be used as messenger RNA, transfer RNA, ribosomal RNA. Messenger RNA is important for, essentially it's a blueprint for creating proteins. Transfer RNA brings amino acids to kind of the ribosomes so the proteins can be made. Ribosomal RNA kind of is within the ribosomes and is the catalyzing region for the protein. So those three types of RNA are all important for essentially making proteins. So you can see kind of how DNA is a blueprint, but DNA can't do it all itself. It needs RNA in order for the DNA to be read and made into proteins. Okay, so today's lecture, again, had a lot of kind of very specific nomenclature involved, had some chemistry concepts that we talked about last time, introduced some new ideas about the macromolecules, lipids, fats, proteins, and nucleic acids. I just threw this slide up here just to kind of remind you what is due the coming week. So make sure you answer study guide questions for chapter 2, but if you're watching this lecture again, watch this lecture. You'll take down your study guide questions, and we're going to meet on Thursday to discuss this material. On Friday by midnight, you should have completed the quiz for chapter 2, and then kind of the following Monday, you'll be taking the adaptive practice for chapter 3, which is going to be due next Monday. I hope you all learned a little something from this lecture. If you have questions, reach out. I'm always happy to answer more of your questions if this material doesn't make sense to you, if you need clarification, if the study guide questions don't make sense to you, if you have questions about the adaptive practice, reach out. I'm happy to answer any and all of your questions that you may have about this material. Okay, that's all I have for you. Have a great day.